Strategies for enhancement in food production. So, day by day the population is increasing in the world. So, to supply for the increased population, the present day food materials are not sufficient. So, to supply here the sufficient food material for the increased population, the conventional methods or traditional methods are not sufficient. So, that is why here we are following the other techniques to produce a large amount of food production. So, here the techniques are plant breeding. The plant breeding. Plant breeding is a genetical improvement of plant or the breeding between the two plants here to produce the superior quality. The plant breeding techniques are generally tissue culture techniques, plant breeding techniques and tissue culture techniques and genetic engineering. So, here different techniques are used to produce the large amounts of food production. Here the plant breeding conventional method, the plant breeding is here the genetical improvement of plants. So, first of all we are going to discuss about plant breeding techniques. So, we are using different techniques to produce large amounts of food production. Here first of all plant breeding. The plant breeding is the production of food materials in large quantities. In the plant breeding, by using plant breeding technique, we produced large amounts of food production in rice, wheat, maize. So, here what is plant breeding? First of all, what is plant breeding? Here the plant breeding is here by using cross hybridization or sexual reproduction. Here the two plants are crossed. The two desirable qualities, two plants are having desirable qualities, the two plants are crossed to produce the superior quality. So, it is by the cross hybridization or sexual reproduction or breeding techniques we can produce a desirable quality or desirable plant with superior qualities. Here what is plant breeding? Here the plants, some plants are having only some desirable qualities like high protein quality, and some plants are having disease resistance. So, these two, some plants are having high protein quality, but low yield and without disease resistance, and some plants are having the disease resistance, but not other qualities. So, here we can incorporate all the qualities, all the desirable qualities into a single variety. So, it is possible with the help of plant breeding. So, genetically we are improving the genetically we are improving the plant. So, as many as possible here we are introducing or incorporating the qualities into a single variety. So, here by plant breeding technique we can enhance the food production. In the plant breeding technique, several steps are there. Here the first step is collection of germplasm or variability, collection of germplasm or variability. So, what is germplasm? Here the germplasm is the collection and preservation of all the varieties, all wild varieties and local varieties in a given crop. If you take one crop or one species, in that species all the varieties are taken and they are preserved. So, the collection of all the available plants are collected, that the collection is called as germplasm. Here variability, variations, here different varieties are different within the same species. Here different varieties are there, wild varieties or local varieties. So all these varieties are collected and preserved, that is called as germplasm. So in the within the germplasm of a given crop or within one species, here all the alleles are present for all alleles. Here all the alleles are present for all genes in a given crop, that is germplasm. So if the here different varieties are present, then only we can here improve the crop, otherwise we cannot. Suppose here if one species is having here only one variety, with one variety we cannot here improve the plant or one species. So here at least here two varieties, the two varieties are present, we can improve the, we can incorporate the two qualities into a single variety. Here the available varieties, all the available varieties are collected and preserved, it is called as germplasm. Germplasm contains all the alleles for all genes in a given crop. So here germplasm collection is a prerequisite, it is important, first important step. So without germplasm we cannot produce the new plant. Here next step is selection and evaluation of parents. Selection and evaluation of parents. Here the here desirable qualities are present in different species. Here number of species are there, but we need some here varieties, some qualities. So the qualities are present in some plants. So the type of plants are selected as parents. So which is having the desirable quality or desirable trait. So the plants are collected and tested. Here if the plants are having in the heterozygous condition, the plants are generally modified or self-pollinated and the plants are modified into homozygous condition, here homozygosity. Some plants are generally self-pollinated and cross-pollinated also. Some plants are only cross-pollinated, they are showing only cross-pollination. Here wherever it is necessary self-pollination and here the plants are self-pollinated, then the plants are modified into homozygous condition. Homozygous condition means here the one gene is having two alleles. Here the two alleles, if the two alleles are same, that condition is called as homozygous condition. Homo means same, homozygous. Here for one gene two alleles are same, this condition is called as homozygous condition. If one gene is having different type of alleles, that is called as heterozygous condition. Here the plants are selected, some plants are selected and they are evaluated. So with desirable qualities, if the plants are having desirable qualities, they are selected as parents. If the parents are in the heterozygous condition, they are modified into homozygous condition. Here after selection and evaluation of parents, these two parents are crossed or cross hybridation or sexual reproduction. Here the pollination is taking place between the two sexual organs. So here the pollination is done between the two sexual organs. The two sexual organs are present on the sometimes same plant and between the two plants also. Here the selection of after selection of parents, the two parents are here crossed. The third step is cross hybridation. Among the selected parents. Here the desirable qualities are present in two parents or two plants. These two plants are to be crossed. Here first one plant is having here male or female. One plant is having here high protein quality. high protein quality, but no disease resistance. This plant is giving low yielding, high protein quality, and some disease resistance is absent. Here it is easily susceptible to diseases. That is why here it is crossed with the disease resistance plant. Disease resistant plant. So, one plant is acting as male plant and other plant is acting as female plant. So, here one plant is having one desirable quality, high protein quality, the male or other plant is having a disease resistance quality. So, these two characters or these two desirable traits are to be incorporated into a single variety. So, here in the female parent only we need female sexual organs. That is in the flower the female sexual organ is a gynoecium. The gynoecium is having number of carpels. Here in the gynoecium or carpel the parts are the swollen basal part is called as ovary style stigma. So, the stigma receives the pollen grains. And in the female parent only we need anthers. But here some plants are having the bisexual flowers. Some plants are having bisexual flowers. Here also it may be bisexual flower. But we need only female sexual organ. Here we need only male sexual organs. Here this plant is one plant in one plant. Here the male sexual organs are collected, or these male sexual organs are only useful. Here the female sexual organ is not useful. Here, but in female plant or male in female plant generally here bisexual in bisexual flower male and female sexual organs are there. So, here there is a chance to occur the self pollination also. That is why here before the opening of flower these here male sexual organs or anthers or stamens completely these here male sexual organs are cut with the help of scissors. Okay, before the opening of flowers or before the production of flowers the male sexual organs are removed with the help of scissors or by using other techniques also. If the flowers are big in size, the anthers are removed or the stamens are removed by scissors. The flowers are very small or the anthers and stamens are removed by hot water treatment. The total flower or inflorescence is dipped into the hot water, then only gynoecium can withstand with high temperature and male sexual organs cannot withstand with high temperatures. That is why here based on the size of flower we use different techniques or methods. Here after the removal of anthers only the flower is having female sexual organ. So, this step is called as emasculation. The removal of anthers from the female parent or flowers present in the female parent. So, from these flowers the male sexual organs are removed. So, the removal of anthers or stamens from the female parent, it is called as emasculation. So, after emasculation, so it is covered with a bag. It is completely covered with a bag. Why the bagging technique is used here? If it is not covered by bag, here the other pollinations or unwanted pollinations also taking place. Here unwanted pollinations, it is useful or desirable pollen grain. It is producing the pollen grains, but here unwanted or other pollen grains also reach the stigma. So, that is why to avoid the unwanted cross pollination, it is covered with a bag. And here from the male parent only we are collecting the pollen grains. So, here the emasculation is carried out in only female parent, not in the male parents. So, we are collecting the pollen grains. So, the pollen grain after collecting the pollen grains, so these pollen grains are dusted or here shifted onto the stigma. So, after removing this bag, these pollen grains are dusted, are transferred. After the transferring the pollen grains, again it is covered with a bag. So, here after the pollination, cross pollination, the cross pollination is taking place between the two here desirable plants or two desirable flowers. Here the plants are having or the parents are having the here desirable qualities. So, after pollination, here fertilization is taking place inside the ovary. Here in the ovules are also here, fertilized. After pollination and fertilization, ovules develop into seeds, ovary develops into fruit. So, it develops into fruit. So, the fruit is having number of seeds. So, based on the number of ovules, the fruits are having the seeds. So, after the production of flower and seeds, so these seeds are collected. So, the seeds are used to raise the next generation. The seeds are giving the new plants. So, it is a cross hybridization among the selected parents. After the selection of parents, the two plants are crossed. It is cross hybridization. And next, fourth step is here testing and selection. Selection and testing of superior or recombinants. So, the recombination means new combination. Here the qualities are present in different plants and they are incorporated into a single variety, single plant or a single organism. So, here after the cross hybridization, here the recombinants. So, here if one plant or the fruit is producing thousands of seeds, all the seeds are developed into new plants, but all these plants are not having the two desirable qualities. So, the probability is very less. So, very less chances are there. So, out of thousands of seeds or thousands of plants, only a few plants are having the two desirable qualities. That is why here the probability is very less. Here it is the selection and testing of superior recombinants. It is very crucial step. The crucial step. Why? Because here the selection is not done properly. So, the total process it becomes waste. So, here all the steps are after all the steps, all the three steps, here the selection is carried out. If the selection is not done properly, so here if improper recombinants are selected, that becomes completely waste. So, here the selection is a crucial step. Selection and testing of superior recombinants. Here superiority or superior recombinants. Superior recombinant means the plants which are having the two qualities. The recombinants are having the both qualities of parents. Parents are not having both qualities. Here one parent is having one quality, other parent is having other quality. So, the two plants are having different qualities, but here the recombinants are having the both the qualities. So, they are superior than its parents. Here superior qualities are selected and they are tested, and these selected recombinants are used for the next generation or here used for the next step. The next step is here testing, release and commercialization. Testing, release and commercialization of recombinants. Here the plants are generally, the recombinant plants are tested in the agricultural universities or agricultural research centers. So, generally in the research centers or research field, the selected recombinants are tested. For example, it is one superior recombinant. The superior recombinant is it is producing a number of seeds. So, from these plants, the plants are grown in agricultural research centers. Why we are growing the here the superior recombinants are grown in the agricultural research centers? Because here the plants are, if the plants are the plants are showing the growth properly when all the requirements are, all the your water, fertilizers, your suitable environmental conditions are provided, the plants grow properly. So in agricultural research centers, here the irrigation means water supply and minerals and here fertilizers, proper fertilizers. So under proper management practices, the plants are grown. Next, from these plants, the seeds are selected, and these selected seeds are grown in the normal or agricultural agri-climatic zones. So in India, different agri-climatic zones are there. So in northern country, here the environmental conditions are different. In southern regions, the environmental conditions are different. The temperature, water availability, so all they are different. That's why here the in these are the superior recombinants are tested in one agricultural center, research center. But here they are showing the proper growth in these agricultural research centers. So but here the same plants are not showing the proper growth, are not showing the two desirable qualities in other regions. That's why it is tested for 3 years. The plants are, the superior recombinants are tested for 3 regions in different agri-climatic zones. For 3 years, the recombinants are tested in the agri-climatic zones throughout India. So the seeds are supplied to farmers. So throughout India, so they are tested for 3 cropping seasons or 3 years. So they are tested. After the test, if the plants are giving, if in all areas throughout India or in all areas, if the plants are giving proper yielding and if they are showing the disease resistance and with high protein quality, so they are supplied to the farmers. Next, they are supplied to the farmers or they are released for the commercialization. They released for commercialization. Here, if the plants are showing proper growth and proper food materials yielding and with high protein quality and disease resistance, the plants are selected or the seeds are collected, and these seeds are certified by National Seed Corporation Limited. So after certification, the seeds are supplied to farmers here throughout the country. So they are released for the commercialization. So here in this plant breeding technique, in the plant breeding technique, here 5 steps are there. In these 5 steps, the first step is germplasm collection of all the variable qualities or different varieties are collected. Next, the second step is here the selected plants are here used as parents. Selected plants are used as parents. The third step is cross hybridation among the selected parents. The cross hybridation is a very time consuming and tedious process. And the fourth step is here the selection and evaluation of superior recombinants. So it is also a very crucial step. And the fifth step is here testing and releasing of superior recombinants. So after the here testing and releasing, here they are released for the commercialization. So it is plant breeding.